I'm Stefano Pagliara, I'm a senior lecturer in microfluidics here at the University of Exeter. In order to study antibiotic resistance here at Exeter, we have developed bespoke microfluidic devices that allow us to isolate and study single bacteria.
What are bacteria? Bacteria exist in all shapes: round, rod-shaped, or with filaments. We cannot see bacteria, but there are around 10 trillion bacteria in us or on us, thus 10 times more than the number of our own cells. These microscopic organisms often measure just 1 micron, 1,000th of a millimetre. Using nutrients from around them, bacteria grow to twice their size and then cut themselves down the middle so that one cell becomes 2, and so they keep growing and dividing. The vast number of bacteria that colonise our body are considered to be an organ in themselves. We call this organ the microbiota. Many bacteria are beneficial for us. These good bacteria digest food and produce essential nutrients for our bodies. What are bacterial infections? Some bacteria are detrimental to our health. They can cause infectious diseases such as meningitis or pneumonia. Bacterial infections are one of the leading causes of death worldwide and are estimated to cause 300 million deaths by the year 2050. Our immune system is responsible for neutralizing bacteria when necessary. However, our defenses can often be breached. For example, when we cut ourselves, bacteria can enter our wound, use our resources, and colonize our body, causing symptoms such as redness, swelling, fever, and fatigue. This is what we call an infection. What are antibiotics? When an infection does not resolve by itself, the physician can prescribe antibiotics. Antibiotics save millions of lives combating infectious diseases by disrupting bacterial metabolism, attacking their DNA, or ripping apart their outer membrane. Penicillins are an example of an antibiotic used for the treatment of sore throats, chest, and skin infections. Antibiotics are generally broad spectrum, affecting many different bacteria, including those that do good to our body. Therefore, antibiotics should be a last resort drug and only be taken when prescribed by a physician. Antibiotics are not effective against viruses. What is antibiotic resistance and tolerance? The misuse of antibiotics is contributing to the emergence and spread of antibiotic resistance. Antibiotic-resistant bacteria continue to grow during drug treatment. When exposed to antibiotics, a few bacteria might change part of their DNA, evolving a genetic mutation that can protect them. For example, by using protein pumps that ejects the antibiotics. These bacteria become resistant to antibiotics, and they can pass on this immunity to their daughter cells or to other bacterial cells. This picture is further complicated by the use of antibiotics in farming, where many animals are routinely given high doses, leading to further antibiotic resistance. However, there are also subtler ways for bacteria to escape antibiotics. Take a population of bacteria with the same genetic makeup, and antibiotics kill the vast majority of susceptible cells, but a very small group of bacteria can tolerate antibiotics and survive. There are 2 types of survivors: persister bacteria that start growing immediately after antibiotic treatment, and sleeper bacteria that do not regrow after treatment. To combat these forms of resistance, new antibiotics must be developed as old ones become inefficient against bacteria. What are microfluidic devices? In order to study antibiotic tolerance, our team at Exeter has used a microfluidic device which enables us to isolate and study single bacteria over time. A microfluidic device is a miniaturized tool containing a tiny volume of fluid, down to 1/1,000,000th of a litre. These devices have recently revolutionized several fields of research, including molecular biology and clinical pathology.
The central part of our devices is that they are constituted of thousands and thousands of small little channels. All these channels allow to study just a few bacteria and are connected to external tubing that allow us to deliver the antibiotics inside the devices. Our findings. We used our bespoke microfluidic device, a fluorescent microscope, and a novel piece of software that we developed at Exeter University. what we discover is that when we dose our bacteria with ampicillin, the majority of those few cells that survive the drug are actually alive, but they are dormant. So we have decided to call them sleeper cells. In fact, despite being dormant, they could wake up anytime and infect humans or animals.
We found that the two types of bacteria surviving antibiotics, sleeper cells and persister cells, have similar features, suggesting the two populations of bacteria are linked. But because sleeper cells are non-growing, standard detection methods cannot differentiate between them and dead cells, giving the false impression that the antibiotics have killed the bacteria.
We then use genetically engineered bacteria that emit light each time a gene is expressed. When we did so, we discovered that sleeper cells produce much less tryptophan, which is an essential amino acid for life, than susceptible cells that are then killed by the antibiotics.
This discovery now allows us to use tryptophan metabolism both as a biomarker to identify antibiotic-tolerant bacteria and as a possible target to eradicate these bacteria. What is membrane transport?
We can also use our microfluidic devices to study membrane transport, which is a fundamental process in bacteria either tolerant or resistant to drugs.
Bacteria need to take up nutrients from the environment through their cell membrane. If we zoom into the surface of a gram-negative bacterium, we see that differently from a gram-positive bacterium, gram-negative bacteria have a double lipid membrane that makes them harder to treat with antibiotics. This double membrane is selectively permeable. Some molecules can cross it, while some others cannot. Lipids, water and gases can cross the membrane via diffusion, moving down from where they are more concentrated to where they are less concentrated. However, most molecules cannot cross the lipid bilayer and are transported through proteins that form pores in the bacterial membrane. Some molecules are transported via channel-facilitated diffusion, a spontaneous passive transport through membrane proteins. Other molecules are transported via secondary active transport, relying upon the electrochemical potential difference of a second transported molecule, or via primary active transport that uses ATP energy. These pathways are often used by antibiotics to reach their target inside bacteria. In order to understand the mechanism underlying these pathways, we use the microfluidic device and a microscope to measure antibiotic molecules entering or exiting individual bacteria.
We are currently developing a mathematical model to understand contribution of the various transport pathways to the accumulation of drug inside the single cells. A mathematical model is an abstract model that uses a mathematical language and a particular set of equations known as ordinary differential equations to describe the system and how this evolves over time. So let's see a simple example. So here I have a cell, a bacterial cell, and I'll use X to denote the concentration of drug inside the cell. I'll use Y to denote the concentration of drug outside the cell. And of course there is transport from outside to inside and vice versa. I can describe this system using a simple differential equation, describing how the concentration of the drug inside the cell changes over time, which is a sum of two parts. Part describing how the drug moves in the cell, and the part describing how the drug moves outside the cell. Now, if you imagine the concentration inside the cell as a function of time, then the dots is what we observe. And using the model, we can predict this increase in the drug inside the cell. So in the long run, we hope that these mathematical models will help us understand how single cells survive antibiotic resistance.
Take-home messages. Over the past 10 years, microbe research has taken off around the globe, engaging teams of scientists from many different disciplines. Some bacteria do good things, some do bad things, but there are billions of bacteria that we have yet to discover.
Our holistic approach for studying membrane transport in sleeper cells will allow in future to discover new beneficial bacteria, but also to develop new antibiotics to fight our daily battle against antimicrobial resistance.